So the final part then of immunity is all about Alexander Fleming, his role in discovering penicillin, making new medicines, and also some little experiments. So this is Alexander Fleming. He discovered penicillin, which is the first antibiotic. Now, here was his original petri dish that he used and led to the discovery of penicillin. So basically, Alexander Fleming was a microbiologist. He grew bacteria in petri dishes and studied how they grew and reproduced. One night before he went home, he basically added bacteria to a petri dish, allowed it to grow, but didn't put the lid on. He came back after a few days, and this is what he found. Now, it would have been very easy for him to just throw this out, but instead what he did is actually notice something about this. He noticed that there was a substance growing here, which turned out to be a fungus. And instead of the bacteria growing all over it, he noticed this area here where the bacteria didn't grow as much. And this was really interesting to him because this hadn't been discovered before. Nothing like this had been seen. So whatever was going on in this area was stopping the bacteria from growing properly in this area. The bacteria here grew normally as expected, but it was this area. This was the area of interest. So what is this and why is the bacteria not growing? A slightly different diagram here. This is what the bacteria should have looked like on the petri dish, but what he found was there was a little fungus here and it created this zone here, or what we call a clear zone, which stopped the bacterial growing. And this is the process that you need to know. So he was growing bacteria on agar plates. Now, agar is basically this jelly-like substance which has all the nutrients for bacteria to grow. So things like amino acids and glucose and moisture. One of the plates had become contaminated with mold or a fungus. He noticed the bacteria did not grow near the mold. So he concluded that the mold produced a substance that must have diffused through the agar and prevented the growth of the bacteria. Now, what I mean by that is this unknown substance must have produced a substance which diffused from an area of high concentration to lower concentration through the agar. As the substance diffused, it prevented the bacteria growing. The fungus then was known as penicillium, and the antibacterial substance was named penicillin, and that was the first antibiotic. Now, the key words that you need to include here if you're talking about this, they were being grown on agar plates. The plates became contaminated with mold or fungus. The bacteria did not grow near the mold. The mold produced a substance that diffused through the agar and stopped the growth of the bacteria. And the fungus was penicillium, and the substance it produces is penicillin. Now, that was Fleming's contribution to it, but it wasn't until much later that they were actually able to purify penicillin and put it into tablet form so that it could be used to treat diseases. It was these two guys, Florey and Chain, who were actually able to isolate and purify the penicillin so that it could be produced on a larger scale. And this is the process that's in place now. So to make penicillin commercially, this is all to do with growing lots and lots of the penicillium fungus, reproducing loads and loads of it. Then when it does produce the penicillin, you extract it, you purify it and package it. So this is the process. They grow the fungus in large fermenters or biodigesters, and they use optimum growing conditions, like for example 24 degrees C, lots of oxygen for respiration, nutrients for growth such as glucose and amino acids, and a slightly alkaline pH. Those are the conditions which the fungus will grow best in. After loads and loads of the fungus has been grown, the penicillin must be extracted, purified, and packaged, and that is known as downstreaming. Only once the fungus has produced it, it's been extracted, purified, and packaged, can it be used by humans to treat bacterial infections. Now, this bad boy shows the biodigester or fermenter. So the fungus is put in here along with the sterile air, which contains lots of oxygen. So the oxygen goes in along with the fungus, the nutrients and the oxygen. So the nutrients that are going to go in is going to be amino acids, because they can be used to make proteins for growth, and glucose, which can be used for respiration. The microbes will grow and they'll reproduce, but when they're growing and reproducing, there's also a lot of respiration that takes place. Now you'll remember that respiration was oxygen plus glucose, which produces carbon dioxide plus water. Now, as well as that, it's also exothermic, which means it releases heat. So if we just left this here with all the fungus on all the nutrients and oxygen, they would just keep reproducing and producing more and more heat. So cold water jacket helps to control the temperature. And remember, we wanted to stay at 24 degrees C. Finally then, the used air which comes out is going to be carbon dioxide mostly, because respiration has taken place. The product, which is penicillin, is then collected through the outlet and purified. And that's how penicillin is produced in a biodigester. Now, that's one example of producing medicines, but new medicines are being discovered and produced all the time. Now, we can't just find a new medicine, package it up and send it out and hope people are okay. There's some main stages which new medicines have to go through. And there's three main ones. There's preclinical trials, which is stage 1. There's stage 2, which is animal testing. And the third stage is clinical trials. So here's what happens in each. Firstly, in the first stage, in vitro testing takes place. Now, in vitro means in glass. So this is where you take the cells, living cells and tissues in the lab, and you test the drugs on them. So you basically want to see if the drugs are harmful to the living cells. It's long-term and expensive because it can be trial and error, and it allows testing before use on living organisms. So for example, let's say we take a little petri dish of cells, we add a drug to it, and all the cells die. You basically need to change the amount of drug which you're using and make up new mixtures. So it can be a lot of trial and error, and it can take a long period of time. Once it passes that stage and you've shown that it actually helps the cells rather than kills them or damages them, it goes to stage 2, which is animal testing. Now the difference is, in the first stage, it was just cells by themselves. Whereas in stage 2, it's tested on animals to check how they work on whole organisms. The benefits are that it avoids testing on humans at that stage. You can check for side effects in living organisms. So let's say, for example, you make a drug to help with some eyesight problem. You might show in this bit that it helps the cells from the eye, but in the second part, you might show that, okay, it might help the cells in the eye, but it actually damages your kidneys. So therefore, you'd need to go back again. The disadvantages of testing on animals is that the animals may react differently to humans, and it also raises ethical issues. Some people may be opposed to testing on animals because they don't have a say and it's against their animal rights. If a new medicine passes in vitro testing and animal testing, they'll go to clinical trials. In clinical trials, the drug is tested on a small number of healthy human volunteers and then patients. So this is basically trying to find out how effective the drug is and finding the optimum dose, and also seeing if there's any side effects. All in all, this process can take years. And the three main stages were testing in a petri dish in a lab, testing on animals, and then clinical trials, which is on humans, healthy humans, and patients. Now, to make sure that labs don't just bring out new drugs and say they're the new wonder drug, scientific research is peer reviewed. Peer review is when new research and new discoveries are scrutinised by other scientists of equal or higher standing to the investigator. The peer reviewers provide detailed feedback and suggest improvements. So let's say, for example, I've developed a drug, I've made it in my lab and I've tested it on cells in a petri dish, I've tested it on animals, and it's gone to clinical reviews. I have to release all of my findings, and then other scientists around the world can either accept or reject the way I've done the study and produced the drug. And they can maybe say that I think you should do this extra piece of research. It's really important because it means that just random drugs which companies want to produce and sell for lots of money don't just make it out to the market, because they could actually be harmful to people.